All right, I think we are all good to go. So before I start, I just want to ask everybody to make sure they're muted. And if you can turn off your video while Kyl's talking, that'd be great. It will help with the bandwidth for his presentation so he's nice and clear. But without further ado, I'm very pleased to virtually introduce Kyhl. I've got to see if I can share my screen here. Hopefully this works. Let's see. Can you see my screen okay?
Yes.
Okay, good. All right. Basically, I first heard of Kyhl a few years back. He just sort of popped out of the ether, out of nowhere, and showed up on Facebook on various mothing groups. And he really stuck out, though, because while there's a lot of us out there identifying moths, we'll get to some where, you know, all the judges, all the experts, they'll just say, give up, that's a difficult group, you can't identify it. But then there'd be this young guy named Kyhl that would, you know, about 3 or 4 hours later, pipe in and say, it's this, here's the reference. And he'd do that over and over and over again, where, you know, everyone else would give up, he would just do it. And I was always impressed with what papers he could find and the amount of research he could do in such a short period of time, and then back up what he said. So I was very keen to get him to come to Cornell and work on moths. Now how do I click through my slides here? All right, we'll have to do it this way. Now, when he first got here, his tastes in cuisine were a little questionable, as you can see on the left. But later on, through the years, he, you know, had a more refined taste. Here's a picture of him in Martinique eating a fresh baguette with some herring and marinade sauce on it. But he has grown up. But in all seriousness, he is phenomenal in the field. Even like on the left here he is in prime blackfly season, where he was miserable, but still he was gung-ho to get out to the tropics, which really he was in his element. Here he is in Guadeloupe or Martinique and tropical paradise. But what was interesting about being in the field with him is he was just always on the go. You know, I usually got to see the back of his head, often from a distance, because he would get out there and he'd find all the good stuff. And not just, you know, field work, he was also really productive in the lab and with papers. Within a month, he had his first draft of this manuscript, which came out shortly after he started here, which was very fast by my standards. Now, it was a bit of a lighter paper, but he's continuously impressed me with how productive he is with papers, including some very difficult ones, even ones sort of beyond the scope of the original thesis. And he's also productive in the field with collecting, and also in the collection, doing a lot of things I never ever asked of him. He curated an immense amount of the collection, and parts that were very difficult to curate that he just, you know, went into and did impressive work. So I'll miss him, but I know he's off to a place that's much, much warmer, because I know he hates the cold here. He's off to the University of Hawaii at Manoa in a few months. Hopefully COVID will let him go, and to continue working on tortricids. And yeah, I'll miss him, but it was a great pleasure working with you for these few years, Kyhl. And with that, I will stop sharing my screen somehow here. Stop share. And hand over the hosting duties to Kyhl.
Perfect. Thanks, Jason, for that introduction. Let's see if I can switch over to my screen real quick. Hold on. Get ready to go. Okay. Share screen. Can you guys see what I'm seeing?
Yep.
Okay, great. Hopefully I can advance the slides. All right, we'll find out. So thanks everybody for coming, and thanks especially to Jason for that introduction. He was much more kind to me in that than he probably could have been. But I just want to say, you know, off the bat, this presentation here is the product of 2 years of hard work here at Cornell. It would not have been possible without the help of a ton of people who I'll mention at the end of this presentation. So with that out of the way, let's just jump right into it. My thesis is broken up into 3 chapters, 2 of which have been published already. But for this talk, I'm breaking things up slightly, a little bit out of order, just for the sake of flow. So here are the 4 chapters. But before I dive right into them, I thought it might be helpful to step back and define a few things first. You might have noticed a couple words in the title you might not be super familiar with. So I want to define a few things first. First, Archipini, what's that? Some of you, after dealing with me at 2 years at Cornell, probably have a general idea of what they are, but a lot of you may not. What's a taxonomic revision? For those of you who aren't as big of taxonomic nerds as I am, which would be pretty difficult, I'm going to try to explain that real quickly, and then also define the geographic scope of my work. So first off, what's Archipini? When I typically meet someone new, say on the bus or on campus or just walking down the street, and I tell them I work with moths, people are always like, Kyhl, moths are so boring. They're the dull, drab, uninteresting, small cousins of butterflies. Why don't you work with butterflies? And I immediately have a whole spiel. Moths are great. They're interesting. They're all sorts of crazy life habits, colorful, big, small. They're everything. And they seem really intrigued. And so they asked me, obviously, okay, Kyhl, tell me about the ones you work with. And then I'll show them a photo of something like this. And the time to take a breath. I can't see you guys, but you're probably laughing right now because this is not a big, beautiful, showy moth. It's admittedly pretty dull looking. But even though externally these moths, these Archipines, are pretty boring to the normal person, they make up for it in terms of the genitalia, which I'll dive into a little bit later. And this will be a running theme. I have a running obsession with moth genitalia, as many of you may know by now. So here we go. This is typical Archipine male genitalia. And the male and female genitalia are 2 of the most important features in kind of circumscribing or defining Archipines as we know them. So on the left here highlighted is typical Argyritinia genitalia. And the defining character for the tribe Archipini, which is what I work with in the males, is, if you guys can see my mouse, Jason, can you see my mouse? Okay, great. Are these apicoventral setae on what's called the uncus here. And the females, the diagnostic feature for the tribe is generally this large hook-shaped cydnum in the corpus bursa, although there are exceptions. So that morphologically are the putative synapomorphies for defining Archipines. But before I dive into anything else, I want to just give a brief overview of Archipini as a whole, because they are an enormously important group, both economically and biodiversity-wise. There's more than 2,000 species worldwide, making them by far the most diverse tribe of tortricity. They have the most numerous, or the highest number of significant pest species, both forest pests and agricultural pests. And despite all this diversity, the tribe Archipini is relatively poorly represented and poorly diverse in the neotropics where I work. So with that out of the way, another question you might have is, what's a taxonomic revision? So I really want to just dive into and tell you about all these species. Before we do that, I need to go over a couple of things first. A brief refresher on taxonomic hierarchies. As some of you may know, family, subfamily, tribe, so on and so forth. The focus of my work is this one tribe in the family Tortricity, which in the Caribbean has 6 genera and 33 species, which at the time of me starting my thesis, I didn't know, but these are the end results. Is now I understand there to be 33 species of Archipines in the Caribbean. So what's a taxonomic revision? Essentially, I am taking all the known specimens, or all the ones I can track down of the group Archipini in the Caribbean, and laying them out on a table and trying to figure out what species are what. If there's any new species, the relationships between previously described species, and just making it organizationally clear for future workers to parse through. So there's a couple of things I can do here after looking at all these species. The most obvious and the most well-known is I can describe something new. So let's say after, poring over these for months and months and months, I think this one might be new. And you might be laughing because, they all kind of look the same. But you have to trust me here that after spending so many times with these mods, so much time with these mods, things become a little more clear than they are initially. So even though this highlighted pair here might look identical to everything else, after a while you learn to spot these small differences. So that's one thing you can do, obviously describe new species. There's another thing you can do here, and you can do what's called propose new synonymies. So let's say you have one species here, species A, and you have different species here, species E. And after looking at them externally, genitalia-wise, other markers you might use to separate species, you come to the conclusion that even though they were described as two separate species, you become convinced that, hey, they're actually the same thing. Like there's no reason to have two separate things, two separate names on what I think is the same species. So I would synonymize those two. So that's another thing you can do with taxonomic revisions. A third major thing you can do is what's called a new combination. And so that's what happens when, let's say I have a species here, which historically, for whatever reason, usually morphological, has been placed in a certain group or certain genus. And after looking at it for a while, I convinced myself that, hey, it actually doesn't belong there. It actually belongs in this other group, in this other genus. And so I would transfer that species from one genus to another. And that's what's called a new combination. So with that in mind, I can jump kind of into the final step of my intro is just the geographic scope, nice and quick. So obviously everyone knows where the Caribbean is, right? But it is still useful to define my research area. So here's the Caribbean, obviously, but I did exclude a couple islands for the sake of simplicity and also kind of biogeographic reasons. So I excluded the Florida Keys, even though some people may refer to them as being, Caribbean. I also excluded Trinidad and Tobago down here, and then islands in the Dutch Antilles like Aruba, Bonaire, and Curacao. So didn't even consider those. Now I can kind of jump into the four main points of my presentation, which represent more or less the three main chapters of my thesis. So the first one deals with treating what are called orphan taxa in Archipini. So this paper was published back in February, I believe, with me and Jason. And essentially what it did is it took John Brown's 2005 checklist of all the tortoises in the world. And he had listed about 7 species as being unplaced to genus. Essentially, they were unknown where they truly belong. They had been placed in a genus before, but everyone kind of had a general idea that they didn't really belong there, and they were just sitting there until someone came along and put them where they belonged in the correct genus. So what I did is I took those 7 species, in addition to 4 or 5 other ones which were obviously misplaced, and kind of figured out where they belong taxonomically, try to find them a forever home, as I like to call it, where they belong with their sister species. And so this is kind of where it started, this whole project, is this specimen collected in Jamaica over 150 years ago, missing its head and missing its abdomen, very clearly didn't belong to the Caribbean fauna based on what I knew at the time about it. This belongs to a genus called Pandemus, which is European and North American in origin. There's a lot of species in Madagascar, Southeast Asia, but nothing in South America, nothing in the Caribbean. So this was probably the product of a mislabeling or an accidental brief introduction, and it didn't really belong there. But unfortunately, the abdomen was gone, the head was gone, and so there wasn't much I could do with it. And it didn't seem appropriate to treat it in a larger revision of the Caribbean. So what I did is, this was one of the 7 species I mentioned that was unknown. I took the other ones and kind of did what I explained with the taxonomic revision, is I took these species and tried to figure out where they belonged. So here's some quick examples. The most exciting thing to come out of this, and was totally unexpected when I started, was we described a new genus. Crotista, Faragona crotista, and Faragona cremnibates were kind of weird taxa that no one had seriously studied in a while. And I had asked the museum, or the British Museum in London, to send me photographs of the specimens and slides, and I was not expecting at all to find what we did. But the genitalia were just so bizarre and so out there and so unknown that it really required the description of a new genus. Here's the second one that you saw in the previous slide, Pteris jamaicana. Turns out is a synonym of this European species that has been introduced to North America as well. Even though I couldn't dissect it for wing pattern, it is pretty unique to the species, and then I'm pretty confident it belongs to that. A third one, just because I think it's really pretty, is, and it's fun to say too, Rhezapoana parana, was unplaced to genus or unknown to genus, and I was able to firmly put it in a new genus, an already described genus, excuse me, resulting in what's called a new combination. And then finally, there were these few taxa which had been placed in Archipini, but as it turns out, didn't belong there at all. Actually belonged in a completely different group called Ateriini. And so there were 3 or 4 of these species that we ended up moving over to that different tribe. And we wouldn't have been able to do that if we hadn't been able to examine the specimens themselves, thanks to people in France and London. And so hopefully that group is in better shape moving forward too, and it's a group that I would love to work with in the future. So nice and quick first chapter. Secondly, we performed like a taxonomic review of the Archipini of the Bahamas, and there were a couple reasons that we separated this from the larger Caribbean revision. This was published actually last year. This is the paper Jason mentioned in his introduction for me. And there were a couple reasons we published this separately. Primarily because, as it turns out, the Archipini fauna of the Bahamas is almost identical to that of Florida, and it didn't seem appropriate to treat it with the Caribbean. With the exception of a single species, which I'll show on the next slide, all of the species that occur in the Bahamas also occur in South Florida. With the exception, here it is, Archipini flavoreticulana, described last year by Jason and I. Externally, a very unique species, something honestly doesn't look like the rest of Archipini, as you'll see in a minute. But the adults here are sexually dimorphic, with the female up top with the brown markings and the male down bottom without those markings. And pretty unique genitalia too for Archipini. In addition to that species, we also redescribed 3 other ones. Archipini kimbali, which is widespread in the southeast US, with a single record from the Bahamas. Amatana, which will come up later, and has quickly become probably my favorite species in the Caribbean. And Clepsis paratana, which is super widespread, occurring from northern Canada down to Central America, just about everywhere. And also turns out occurs in Cuba, which I'll mention later. In addition to redescribing the species, I also performed a quick literature review on the known host plants for the species for which host plants were known. And that was also included in that publication. The third chapter, and honestly the biggest chapter, is the one I'm going to talk about now. It's currently in review at Zotes, hopefully to be published later this year. But this is the bulk of my thesis, and it's been a lot of joy to work with, but also a lot of frustration at times. So this is, in essence, the result of my entire work at Cornell. 2010 was the most recent paper that dealt with any of these species, and then 2020 is where we are now. And so even though it doesn't look like, for example, Archipini, there are many new species, this is really a pretty gross simplification based on how many taxonomic kind of changes I had to make, synonymies, combinations, in addition to new species. So what my goal here is kind of to introduce you to some of the species I've described or will be describing, it's not technically published yet, and then share 1 or 2, I think, fun facts about each of them. I've sat through some talks like this before where people are just listing, here's species A, this is what separates it. Here's species B, this is what separates it. And no one, frankly, wants to listen to that. And I know that's the case for you guys. So again, I'm just going to try to show you some cool, pretty photos. Hopefully you'll fall in love with these moths as much as I have. Before I do that, though, I want to just explain my rationale for describing these new species, or for transferring species from one genus to another, or proposing new synonymies. Four-wing pattern obviously is a huge indicator of species boundaries in Lepidoptera. So that was a primary character used. Like I mentioned before, the male genitalia, especially in certain genera like Archipini and Cladocaria, is extremely useful. And then for other genera, such as Clepsis, the female genitalia is actually more informative. And so I dealt with all of that. In addition to kind of morphological data, distribution also turns out to be hyper useful. A lot of these species, with the exception of Amatana and a few of the coastal species, are highly restricted in their range, only occurring on a single mountain peak or a series of closely situated peaks, like you can see in the figure here on the left. But also included a little bit of molecular data too. So for a few species, we weren't able to get barcodes, but for the majority, we were able to get Cytochrome oxidase 1 barcode data, and that was used to, one, associate sexes, because a lot of these species are super sexually dimorphic and difficult to associate males and females. But two, also to kind of circumscribe species boundaries, which for a couple of these were problematic. In addition to that, I went ahead and redescribed all but one of the Caribbean species. There's one species, which I'll explain in a second, we weren't able to locate the holotype, and so we weren't able to redescribe it. But Rosowski, who was the primary worker in the Caribbean and also the neotropics for basically all tortricids, left a lot to be desired in his descriptions. So here's just an example I pulled out from one of his publications from one of the Caribbean species. It's essentially a 2-sentence description. The genitalia are figured, but there's no adult photograph. So you have to guess what it looked like based on this 2-sentence description. And honestly, you saw how similar all these moths are. They all kind of sound the same. So I also redescribed all the Caribbean species minus one. And finally, in order to aid future identification efforts by other Lepidopterists, I also developed keys for all the genera and all the species occurring in the Caribbean. So here's just a snapshot of a couple of the keys I've worked on, primarily based on genitalia, but also range and four-wing pattern. So with that out of the way, I'd like to introduce you to some of the new species I'm describing, starting with probably the weirdest group of Archipines. I'll explain why I did quotes here in a second. The weirdest group of Archipines in the Caribbean, the Myticichia group of genera. So most moths, as you probably know, are nocturnal, pretty drab colored, as much as I hate to admit it, and pretty difficult to tell apart. The Myticichia group of genera are these bizarre diurnal, oftentimes I think jumping spider mimicking moths found throughout the neotropics. And they've all, I think, I'll explain here in a second, converged on a similar wing pattern and behaviors. The morphology and preliminary molecular data for these, in my opinion, suggests that they are very, very distantly related, if that. Several of these genera might be polyphyletic, and all have just converged on this similar wing pattern. For the sake of continuity, though, I have decided to treat these as Archipines going forward. So again, just absolutely gorgeous moths. I'll let you just stare at these for a couple seconds because I am completely enamored with these, as I hope you are too. So the primary reason these guys have been kept with the Archipines as of late is because of a really simple structure in the female genitalia. Like I mentioned earlier, the Archipines are defined by a hook-like or J-shaped sydenum in the female genitalia, like you can see here. And most of these guys have that. But when you look at the male genitalia, they're just all over the place. So like this one here, it's, for example, has this really well-developed uncus sticking out here, really densely spined socii, whereas this other species, it's the complete opposite. It doesn't have this well-developed uncus. The valves are, excuse me, completely different shape. And in my opinion, it's pretty clear that these guys are very, very distantly related and have been artificially kind of lumped together just for the sake of simplicity. I'd love to, again, work with these in the future, but there's only so much you can do in 2 years at Cornell. So here's this group as I define it in the Caribbean. 5 species, 2 of them are new. Santa Romana was described from Brazil. I'm also reporting it for the first time from Grenada. Myticichia cubi, even though it's described from Cuba, turns out is super widespread. I have records from Hispaniola, Honduras, and Costa Rica for the first time. Myticichia nyalinda, which I'm naming after my grandparents, Niall and Linda, photo down here, is also pretty widespread, but doesn't occur in Central America, just on Cuba and Hispaniola, and is externally very similar to cubi. Myticichia jamaicana, unfortunately, I wasn't able to examine. It probably doesn't actually belong in Myticichia. I think it belongs in Mythochromosis, but I couldn't examine it, so I can't make that decision for sure. And this gorgeous moth off to the right is Mythochromosis lesliae, named after my mom in the photo here. So I can't, as soon as I saw it, and as soon as I saw it was something new, I could not name it after my mom. Another weird thing about this group is, with the exception of cubi, which has a couple high elevation records, all of these genera are coastal things. I mean, 10, 20 meters off the coast, basically on the beachfront. Another kind of indicator that these aren't archipines. Clepsis is probably the most frustrating genus for me. I think for a lot of torturistic taxonomists, this is true. The male genitalia are essentially all identical. They all look the same. Forwarding pattern is all the same. But the female genitalia, with a few exceptions, is pretty distinctive. And so I was able to use female genitalia for this group to identify 4 new species in the Caribbean and 1 new record from Cuba. I had mentioned Paratana being found in the Bahamas. Here we also reported from Cuba for the first time. So these 4 species are named after people who have meant a lot to me in my life, either professionally or personally. So Darren, the top one here, A and B, after my father. James Stewart, who, James Stewart, who passed away in December. Davis after Don Davis of the Smithsonian, who collected the majority of the type series of this species on Dominica. And I was actually able to see in real life in Guadeloupe. This is a live photo of that new species on Guadeloupe from this past summer. And then Clepsis peroniae, after a professor at Davidson who passed away unexpectedly in December, who also meant a lot to me. Peroniae is unique in that it's only known from the female holotype, 1 specimen, and that's it. Sorry, I didn't expect to kind of tear up like that. Again, these ones, with the exception of Paratana, which is, if you remember from previous slides, is super widespread in North and Central America. With the exception of that one, all of these are also high elevation things. James Stewart being the most widespread and common on Hispaniola, and molecular data suggests it might actually be 2 species, but without any morphological characteristics to back that up, we've decided to just keep it as 1. So Archipini is the most diverse genus in the Caribbean by far. And molecular data, in addition to subtle morphological characteristics, suggests there are at least 4 distinct groups of Archipini present in the Caribbean. And so I'm going to walk you through group by group each species, and again tell you 1 or 2 interesting things about each. So Archipini amatana, that along with J. McCanna and Vinalesiae, form a distinct group of coastal, low elevation, probably polyphagous moths, externally all very similar, genitalia-wise also very similar, but molecularly pretty distinct. Flava reticulana also probably belongs to this group, as does Kimbali, but again, molecular data is lacking for those two species. Amatana, I've mentioned a lot before, has quickly become probably my favorite species in the Caribbean, just because of how hyper variable it is. So all 8 moths in this diagram here are a single species. That's all Amatana. It occurs in the Cayman Islands. It occurs Bahamas, Cuba, Jamaica, Hispaniola, Florida, low elevation, high elevation. It is probably the most common, most widespread species in the Caribbean basin. J. McCanna here, G and H, to use my mouse more, is restricted to, as you can probably guess, Jamaica. It seems to replace Amatana at mid to high elevations on that island. And here's a photo of that species. The first, probably only live photo of the species was posted on iNaturalist about a month ago, and I freaked out when I saw it because I was like, I know what that is. I re-described it. And I was able to put an identification on that. Vinalesiae is kind of a weird runty looking thing, only known from a single location on Cuba, but molecularly pretty distinct from the previous two. The other group I want to quickly mention here is Archipini ceramica. And ceramica is an oddball in the Caribbean archipine fauna. The genitalia are a much better match for what's called the Ponera group of central Mexico. Very distinct genitalia, unlike anything else in the Caribbean. So here's a couple distribution maps for these species. Like I said, Amatana, hyper variable, hyper widespread, just everywhere. The other one's pretty restricted in their range. Ceramica, pretty widespread on both Hispaniola and Cuba. But because I wasn't able to find any morphological differences between these two taxa, Ceramica and Grand Piedre, which were previously separate species, I've decided to sync Grand Piedre to a subspecies of Ceramica. There's molecular differences, but I couldn't find any morphological differences. This next group of Argyrotinia I think is equally fascinating. This is, as far as I can tell, based on morphology and molecular data, a exclusively Hispaniolan radiation of Argyrotinia. So these 6 species, 3 of them have been previously described, 3 of them I'm describing as new, are restricted entirely to the island of Hispaniola. Externally, they're pretty distinct. Male genitalia are pretty identical, female genitalia are pretty identical. So most of these are based on 4-wing pattern, and then subtle characters in the male genitalia. So here's Bisignata is A through D, pretty restricted in its range, only found in the southernmost mountain range of Hispaniola called Sierra de Bajaruco. Meanwhile, Felicana, which is its probable sister species, is super widespread and essentially everywhere else in the Dominican Republic and probably Haiti. So you can see the maps here. Bisignata pretty restricted, Felicana pretty widespread. Argyrotinia nuizana and rosowskiana are also members of this group, but these are essentially restricted to single mountain peaks on Hispaniola. Nuizana is distinct externally, just bizarre genitalia for this group. And rosowskiana, G and H here, pretty uniform, nothing stands out about it, but this is being named after Joseph Rosowski, who is a Polish entomologist who has worked extensively in the neotropics and has arguably, actually not arguably, factually described more tortricid moths than anyone else in the world. 2 other new species are Argyrotinia cryptica, which I'm describing 2 separate subspecies based on subtle morphological characters and then range distribution. And then Paradisei, I'm naming after Chris Paradise of Davidson College, who is my undergraduate advisor during my time there. That's E and F. And the cool thing about this kind of species pair, they're sister based on morphology and also molecular data, but they have these really neat organized distribution. So each of these species or subspecies is restricted to a single mountain range on Hispaniola. And exactly how they came to this distribution, I don't quite know, but I'd love to investigate further in the future. The final group of Archipini in the Caribbean are these kind of oddballs that don't quite fit with anything else I've seen in Central or South America. Archipini cubi, which again, running theme here, even though it's named after Cuba, isn't restricted to Cuba. I'm also reporting it from Hispaniola for the first time. And then Archipini browni, which is E and F here, naming after Richard Brown of Mississippi State, who is a big mentor to me as a lepidopterist, working there for 2 summers. Just again, distribution maps of these 2 species real quick. Cladoncaria is probably my favorite group in the Caribbean, and you'll quickly see why here. Molecular data suggests they might just be a highly derived lineage of Archipini, but morphologically they're just bizarre, and you'll see why here in a second. I hope I'm hyping this up enough. Like, these guys are just so freaking cool. So I'm just going to jump into the names real quick, and then I'll dive into my 4th chapter, which deals with a unique coupling mechanism with these. So I've split these up into 2 groups based on morphology, and again, I'll explain that in a later slide. I'm really building up the anticipation. I know you're just chomping at the bit to hear about that. But the Acroclina group, 2 previously described species and 2 new species, both from Hispaniola. So Cladoncaria rawlinsana, named after John Rawlins of the Carnegie Museum, who collected the vast majority of all of the material I've worked with during my time at Cornell. Led countless expeditions to the Dominican Republic and collected, honestly, probably 75 or 80% of the moths I've worked with. So that's in honor of him. And then Cladoncaria praedictana, which is G down here. You can probably guess where that name comes from, predicted. And again, you'll see why on a next slide. I'm really just building the anticipation. I can tell you guys are super excited, even though I can't see you. And this other group, the Mesosygnaria group, only 1 new species, Cladoncaria taino, which I'm naming after the principal inhabitants of Hispaniola prior to European colonization, only known from the males. So another kind of neat thing about these is, with the exception of Maestrana, which is kind of widespread on high elevations in Cuba, all of these species are restricted to essentially a single mountain peak. Mesosygnaria is a series of peaks, but all the other ones is essentially a single mountain peak. It might be an artifact of collecting because so few specimens of these species are known, or it also might just be these species are rare. I'd love to go down there and try to collect some. So that leads me to my 4th kind of topic in this presentation, checking time, making sure we're good. In this new coupling mechanism I've uncovered in this genus Cladoncaria. So before I do that, I need to kind of give some background on typical Archipine genitalia. So this is 6 different species of typical Archipini genitalia. And as you can tell, they're all remarkably identical. After dealing with them for 2 years, I have been able to spot certain subtle characteristics, but even then it's frustrating how few differences there are between some of these externally very distinct species. Meanwhile, this is Cladoncaria. They're just, excuse me, morphologically bizarre. They have this one group up here with this bifid uncus. You see how it splits off into like 2 different forking branches. It's called the uncus. And this other group, which doesn't have that split, but just huge apically widened uncus. And so as soon as I saw these, I'm like, holy crap, like what are these used for? It has to have some sort of, function. And it wasn't really until I started digging into the literature on coupling mechanisms that I started to have a feel for what these might be used for. So before I dive into that, I want to just give you a quick overview of kind of Lepidoptera genitalia. I'm super excited about this topic. So bare bones, this is what happens when mating takes place. The two structures in the female genitalia that you need to keep an eye on are what's called the papillae anales, which are pointed out here, and the ostium here. So this is its resting position. This is just a normal female moth just flying around. This is what typically happens. During copulation, while the moths are mating, what happens is somehow different structures interact with different things to push the papillae anales into the abdomen. You can't generalize and say this structure in the male always corresponds to this structure in the female, because Lepidoptera genitalia are just so wildly variable. But more or less, this is what generally happens for most species. Some structure in the male genitalia pushes the papillae anales into the abdominal cavity of the female, thereby simultaneously exposing the ostium, which is where copulation actually takes place. So push the papillae anales in, that simultaneously opens the ostium for mating. Here's one example of this weird species described from southern South America just a few years ago, of this bizarre, unique coupling mechanism at the time, where the male uncus, let's see if I can do it here, the male uncus hooks into this little pocket outside the genitalia itself, but on the top of the abdomen, hooks in this little pocket. Simultaneously, the gnathos, which is this kind of hook-like structure here, slides in between the papillae anales to assist with opening up the ostium. So that's just one example I found in the literature. There are very, very few examples of this where it's been demonstrated, but this is one of the better examples I've found. So with Cladocardia, like I mentioned, there are two groups. So there's this one group, Acroclena, which has this bifid uncus, the kind of forked split uncus. And it also has, it's difficult to see here, but you'll see in future slides, this bizarre kind of vertically bifid terminal plate of the gnathos, which I'll explain what that does here in a second too. So that's that one group, keep that in mind, Acroclena group. The Mesocygnaria group, very different. So the uncus, like I mentioned before, is just apically broadened, kind of triangular, and it doesn't have that split or bifid terminal plate of the gnathos. The females also neatly split into these two different groups. So the Acroclena group females have kind of a notch in the side of the papillae anales. They also have a little groove here. It's difficult to see in this specimen, but it'll be more clear in future slides. A groove in the end of the papillae anales. And you might be thinking, that is interesting. Like, what could that correspond to in the male? Meanwhile, the Mesocygnaria group, which if you remember from the previous slide, has that broad uncus. Females of this group have just these massively swollen papillae anales, like way bigger than any other Archipini I've ever seen. So here's another example of a different species. Again, this is Acroclena. This is known from Hispaniola. Pretty narrow, rounded arms of the uncus, and correspondingly narrow grooves in the papillae anales. It also has this asymmetrical terminal plate of the gnathos. It's more clear. You can see here the weird shape here than the previous slide. And I should also mention at this point that these specimens have been artificially flattened under glass. This is not how they look in real life. You know, the arms coming off like this. In real life, I'll show in a later slide, they kind of rest like this. So again, here's another species. This is Rawlinsana. This is a new species also from Hispaniola. The arms of the uncus are quadrate. They're kind of squared off and kind of a more symmetrical terminal plate of the gnathos. And if you look at the female, it has correspondingly larger, more square grooves in the papillae anales. And the terminal plate of the gnathos, I was, for the longest time, trying to figure out exactly what that could possibly correspond to in the female genitalia. And after a lot of looking and a lot of searching, I noticed that there's this small little pocket in between the papillae anales. It's not super visible in this specimen. It's really difficult to observe if you press the specimens flat. But when you still have them floating in ethanol, kind of three-dimensional structures, it's pretty clear that there's a little pocket there. So like I mentioned before, those specimens were pressed flat under glass. Here's what the genitalia looks like externally in real life. I brushed some scales off the end of the abdomen so you can clearly see what's going on. But essentially what I think happens is the uncus, these two arms here, fit pretty neatly into the grooves of the papillae anales here in the female. And these kind of cup-shaped papillae anales are unique in, as far as I can tell, all of tortricids. Most all other tortricids, honestly, the defining character for tortricids as a family is kind of flat pad-like papillae anales, two-dimensional structures. So to find tortricid with these radically modified papillae anales really brings up a lot of questions about its function, whether it's a novel ovipositional strategy, or it's a sexual arms race between the species, or something else entirely. I don't know, but it doesn't take too much imagination, in my opinion, to see how these two structures in both the male and the female genitalia could fit together in like a little puzzle to essentially push in the papillae anales, expose the ostium, and mating takes place. So I think I'm just right about on time, so I'm pretty happy with that. Just to summarize, 33 species in the Caribbean, 32 are given detailed diagnoses, countless new species, new combinations, synonymies. This represents the first complete revision of a tortricid tribe in the Caribbean. And I've also uncovered an entirely new sexual coupling mechanism in Lepidoptera. Something like this has not been seen before to my knowledge. And with that out of the way, I cannot thank enough this whole list of people, but especially Jason for all the work he's done as both my advisor and mentor, both personally and academically here at Cornell. Jim Liebherr for all of his work with taxonomic kind of weird minutiae, just all his help. And then also Rob Raguso is also on my committee, provided a lot of literature to get me started when I first moved up to Ithaca. A whole bunch of other people who I've listed here, I can't name them all, but have all helped in one way or another. Friends and family, NSF grants, and certain Cornell specific fellowships have also helped me along the way. So literature cited, and with that out of the way, I think we have like 10 or so minutes for questions. I'll switch the admin back to Jason so he can let people ask stuff.
All right, thanks so much, Kyhl. I think to keep this simple so that we're not all talking over each other for people that have questions, if you want to ask a question, turn your video on, and then unmute yourself and pipe in, and then we'll be able to see the lineup of who has questions. So any questions?
Uncus. I like the word uncus. I just wanted to say that.
Thanks, I do too. There's a lot of fun terminology I left out that I could have said, but.
Wait, what's another one?
Oh boy, Nathos, I mentioned that. Tegumen is a fun one to say.
I like that.
Saculus is a fun one. I like that as well.
Yes, saculus. I like apically widened uncus. I needed to say that. So great job also.
Thank you.
Hey, Kyhl, I have a question.
Sure.
Really nice seminar and a ton of work. I have a question and then a comment. So could you talk a little bit about the host plant associations of these moths? Is that perhaps are they narrowed or highly specific in their host plant preferences, and might that explain?
So I can't talk too much about the Caribbean fauna, unfortunately, because so little is known. I can talk a little bit about the North American fauna because that's known a little bit better. The Amatana group, which I mentioned, consists of Amatana, Jamaica, and Vinalesii, are all probably polyphagous. Amatana has been recorded from something like 30, 40 different species of plants. Jamaica is the only species of exclusively Caribbean moth, which I have a host plant record for. It's a weedy aster. The rest of the Archipines are all high elevation in the Caribbean. Most of those are probably pine or podocarpus feeders based on possible relatives in Central and South America. But again, nothing unfortunately is known beyond the Amatana group of host plant associations.
Okay. So it's really the high elevation that puts limits on their geographic distribution.
Most likely, yeah.
Okay. And then my one other question would be is so if I understood from your phylogeny, Cladoncaria, which is the really cool group with the really bizarre genitalia, comes out within Argyrotinia.
Yeah, it nests within Argyrotinia, but that's just based on Cytochromatosidase 1. It's a very preliminary phylogeny.
Okay.
I've elected to keep it as its own genus until, you know, more robust molecular work or morphological work can be done.
Okay, I see. Right. Just wanted to check on that.
Yeah, no, it does come out as within Argyrotinia.
Okay. Great, thanks.
I have some comments and also a question.
Okay.
First, I want to say that was a really great presentation, Kyhl, and I'm very proud of you and what you've done in the past 2 years. And you also made me cry as well. So I think it's really great that you're naming all of these awesome moths after people in your life, and I'm really happy that you named one after James too. I think he would have really liked that. I think it's a really great way to honor him. So I wanted to thank you for that. And excuse me, because I don't know anything really about these moths other than just from being around you. So this might be a not very in-depth question, but if you had to estimate what percentage of Archipini moths have been described versus the ones that are probably there? Like, how far have we been able to actually know what the scope of how many species are in the Caribbean of Archipines?
Oh, okay. You're talking about just the Caribbean. Yeah. So I have a feeling that at least on Hispaniola, the vast majority have been described. There's a few where either the male or female is unknown, and that's kind of a missing piece. I think Cuba is really a black box in terms of Archipine diversity. I don't think when all is said and done, if everything is ever figured out completely, it won't have the same numbers as Hispaniola. But I think as it currently stands, there's only 6 species known from Cuba. I wouldn't be surprised if there's 4 or 5 more known from high elevations. It's just getting down there and collecting is so difficult for so many obvious political reasons historically.
Thank you.
Hey, Kyhl, I have a quick question. It was related to your talk, which was awesome. I loved it. More about what you're planning on doing in Hawaii in the future.
Sure. So I guess for those of you who don't know, I am hopefully moving to Hawaii in August to start a PhD under Dan Rubinoff. A lot of the details have yet to be fleshed out, but what most likely will happen is I'll work with developing molecular tools to identify tortricid pest species being intercepted in Florida and also in the US through USDA funding. And then also hopefully my real passion lies in taxonomic revisions and hopefully doing some sort of taxonomic revision of the tortricid fauna of Hawaii. So I guess kind of two goals there.
Cool, thanks.
We have time for one more quick question. Is there any questions out there?
Can I jump in and just say that Dan Rubinoff made the right choice taking on Kyhl, because he's going to be able to do a dual program, one in an applied side and one with basic revisionary systematics, probably they have a nice molecular lab. You'll have all the tools you need. But we in the collection had Kyhl as our GRA on our NSF grant, which is officially done. And if you look on YouTube, maybe Kyhl can send the link. He's got 6 videos for aspiring lepidopterists to learn how to deal with moths from the field to the pinning block to the pinned specimen box. So Jason and I are just ecstatic about how well he did that. It's a really fine piece of work, and it puts the collection in a really good light. So thank you.
Thanks.
Can I ask one question?
Of course.
Yeah.
First off, Kyhl, excellent job. Really excellent work. So I was really thrilled to see that some of the skills you learned in the phylogenetics class made it into your thesis. Were there any big surprises when you sort of analyzed your data and looked at it in this phylogenetic context or in the biogeographic context?
The biggest surprise for me, like I mentioned to Brian briefly, is the fact that Claudine Carré comes out as nesting within Archipini, which if you ask Jason, he knew that coming in. Like that's what which I'm like, okay, Jason, yeah, of course you knew that. But based on the morphology, I had no idea that was going to happen. And again, it's very preliminary molecular work, and I think it just really encourages more work needs to be done. But that for me was the biggest surprise. You can probably see that based on the morphology, they look nothing alike.
All right. Well, I think we'll stop there. Thank you so much for coming out, everyone. It means a lot to Kyhl and myself. And thank you, Kyhl, for such an excellent presentation. Sorry, I forgot to mention the videos. They are really good. Do check them out. They're linked to on our CIC Facebook page. They're on YouTube, and they are well worth the watch if you want to see the tedious work that Kyhl has done over the last 2 years. And yes, thanks everyone for coming out. Have a great day in quarantine.
Can we everyone unmute and give him a round of applause?
Congratulations.
Great job.
Everybody.
Great job. Yeah.
Great job, Kyhl.
Congrats, Kyhl.
We love you.
Yep.
Yep.
Fantastic. Fantastic.
Good job, Kyhl.
Yeah.
Knew you could do it.
I do that. Hey, Kyhl, it's Aunt Kim and Eve. Congratulations. That was awesome. We're so proud of you.
Thank you. I didn't see your name there.
You've got quite a fan club.
Work, man. Proud of you.
Oh, thanks, Ray.
Great job, Kyhl. You made me cry.
Oh, that wasn't my goal. I'm sorry.
No, I mean, it was so sweet for you to name the species after your mom and your grandparents, but I forgot about James. What a lovely way to honor him. So, thank you. Very proud of you. All right. Bye, honey.
Bye.
Bye.
Bye, Kyhl.
Bye.
It stopped snowing in Ithaca.
Shouldn't be any more snow in Ithaca. You're safe now.
Give me a second. No, hold on.
So Kyhl, did you get the invitation from me for like 11:10? Did that show up on your email?
I haven't. I got the notification. I haven't checked my email yet.
Okay, that's fine. Let's try that one. I don't know that that Stephanie sent us a separate link for just the 4 of us.
Okay. Do you mind if everyone just takes a quick 5-minute bathroom break?
Absolutely.
Yes, please.
Okay.
It is time. Seeing as I've got the invite, when I get back, I'll open it up and we'll see if that works. And if not, we can talk by phone or email and get it together. No worries.
Sounds good. We'll figure it out.
Not a hurry. Great job.
So Jim, just to clarify, you sent an email recently with a new link?
I did at the time when I looked at Stephanie's email and saw that the link for both we got 2 links for the seminar, but no link for a separate thesis defense. And then I put in an invitation. Unfortunately, because we blocked out, I believe, 10 to 12 for this meeting, I couldn't change the time for the restricted meeting with just the 4 of us to anything earlier than noon. At this point, before I go to the bathroom, I will go back to that Zoom meeting and see if I can change the time to something like 11:15. Okay?
Okay, sounds good.
And then I'll send an email. So.
All right, sounds good.
Great.
Let me give it a shot.
Sounds good. See you soon.
Great job, Kyhl. Yeah.
Thanks. Bye.